Ladies and gentlemen, Dr. David Stojdl. So we're going to edit that together later. So you mentioned this idea of using biology to fight biology, and CHEO discovered, now here's another test of my pronunciation, Maraba virus? Maraba? What is the Maraba?
It's an insect virus, really, that was isolated in a mining area in Brazil. We didn't isolate it. It's in a library, biological library down in Texas, and we just basically took it out of the library and tested it here. So
took the virus out of the library.
Checked it out, yeah. Yeah, so we just rode away, and we got 20 or 30 of these viruses. Because we already knew from previous work we did that this particular family of viruses has this property that can sense the genetic difference between a tumor cell and a normal cell. And that was something we felt was the basis of a good therapeutic for cancer. So the way that we tend to treat cancer now is to try to poison the DNA inside of that cell, mostly, in terms of this thing that kills patients most, that disseminates disease. And that's a concept that's 60 or 70 years old now. It served us incredibly well, but there's too often situations where it doesn't work. And there's also a lot of side effects associated with that, especially being from the kids' hospital, is really important for children. Because children will have to deal with some of these side effects later on in their lives. So we thought that that was a better starting point, something that was biological, something that could sense the difference in digital terms between the target and the things, the rest of the body that one would leave alone. And then we spent the next 15 years figuring out why that was. And, well, how long do you have?
In the next 45 seconds, what did you do?
Well, I guess one of the things that is different between a tumor cell and a normal cell is there's a bunch of signaling pathways inside a cell that will tell that cell it's okay to divide, it's not okay to die. And these types of signals get crossed in a tumor cell because it's not interested in stopping its division, and it certainly doesn't want to die when it's been told to sacrifice itself. Many of the same wiring that goes on to make those decisions is also involved in protecting cells from virus infections. Just happens to be cross-wired. And by sheer accident, no real bathtub eureka moment. In a sense, it was a chocolate and peanut butter type of scenario where we were working on understanding how cancer worked, and we started to realize that many of these same things that we were interested in were also things that protected us from viruses, and so we put the two things together, and that's how it started.
So, moving forward then, I mean, once you've almost had a relationship with this virus for 15 years, I'm wondering, what's the next stage?
Well, as you heard in some of these other videos, the critically important thing is that not that we really understand how things work in the lab, but that they actually matter in the clinic. And so we've spent quite a bit of time, myself and my colleagues, trying to take these ideas that we sort of validated in sort of the models that we can in the lab, and get them into patients. And so, after 15 years, something I thought would only take a couple years as a trainee, but take a couple years, put this in the clinic, and then we'll know. And then if it doesn't work, then I can go sell textbooks or something. And it's taken 15 years to get there, but now we're actually in a trial. We're testing this virus in people. So, hopefully in a year or so, we'll know whether it was all.
Is the timeline really that close that it's within a year?
I think so. We'll start doing
We'll sell the textbooks just yet. They still need a year. But that is an interesting point, though, just the refocusing of anybody who's working in a field like this. Your own timeline for how long. I don't know how you ever do think, oh, this will take a year, this will take 4 years, this will take 8 years. Do you have to constantly be changing your own expectations?
Yeah, I think it's important to be able to delude yourself as much as possible. I think if you couldn't, then you'd probably just not start. So I think that's a skill I have. I'm honing it up. But I think it's important. It's a bit off topic, but I think one of the skills of being, an effective scientist is to always be able to dream. Make sure your dreams are a little bit more enticing than all of the roadblocks that you see in front of you. And I think that's what really ends up being the success, at least the success is persevering when it looks impossible.
How do you do that, though?
It's a work in progress. I think people do it different ways. It's, people have different emotional makeups. Me personally, I don't know. This is one way. I think I don't actually do a really great job of it. There's a lot of emotional ups and downs, and I think, feeling as if I perhaps was lucky. We started to very early seeing really promising results. So that got us feeling like, hey, we may actually be able to do something here. As a student, when I was training to do something about cancer, it was a really strange training in that it was right from the get-go, we were sort of trained that really it's not feasible to cure cancer. It's something that we can maybe try to help patients manage. It was, I felt being quite naive, fairly defeatist type of thing. And that sort of persevered for a long time. But now I think that totally changed. I think now, being a trainee, if I was walking in the lab now, the atmosphere is very different. There is a real feeling that this is a puzzle that can be certainly solved. It's being solved, sort of one indication at a time. And we're onto a very different field for how to treat the disorder. It's not necessarily about finding a drug that will kill the cancer cell. It's more about retraining our own immune system to be able to find the tumor and deal with it using the most sophisticated weaponry that we know of, which is actually our own immune system. So that's where the field is going. That's where our own work is going in many other labs here at CHEO and around the world.
Okay, let's see what you do next. Thank you so much for everything you're doing.